All right, lecture 26. Last lecture we talked about the female reproductive tract, the development, the preparation for fertilization. Now let's bring the sperm and ovum together and produce a fertilization. If you look on plate 156, kind of overview, here's the uterus, there's the cervix. So the cervix then delineates the separation from the vaginal vault and the uterus itself. These are the uterine horns. In the anatomy of our domestic animals, they vary from species to species. Dr. Fondel will make you memorize all of those. Let's view it generically here. This is the fallopian tube, or what in your text is referred to as the uterine tube. And this is a long extension out of the uterus that sweeps out into the abdominal cavity. It is a tube lined, so it has cells lining it, but at the very end, it's open to the abdominal cavity. It is characterized by an enlargement and variegations or fingers, what are called fimbria. These fimbria are motile. They tend to move, and it is their role to sweep a released ovum into the uterus. So the ovary sits physically distinct from the uterus. They're not connected by a tube. So there is a space between the ovary and the uterine tube, a space in which there is the potential for this ovum to get out. If this ovum gets fertilized right up here in the opening, up in the fimbria, so if a sperm manages to make it up here, fertilize this egg, and the egg drops out, the egg will tend to attach to the abdominal wall. Well, the abdominal wall has vascular blood supply, and in the early developing embryo, there's adequate amounts of oxygen and substrates diffusing throughout the abdomen and along the abdominal wall to support the development of that embryo. The embryo will start to grow. That is called an ectopic pregnancy. Anytime you see the word ectopic, that means it is occurring in an abnormal place. Not good. So you want this egg, this ovum, to be swept up and then to meet with a sperm, usually somewhere along this uterine tube. These numbers are averages for humans. They vary from our domestic species, but you just have kind of a concept that you may start out with 300 million. You may, and in cattle it may be on the order of billions of cells that start here. And they all swim with a purpose. Now, it's not a particularly organized swim. They don't have eyeballs. And so they tend to move in this direction, but you can see that there's a significant degradation in terms of the numbers that actually reach the uterine tube. So one of the things that you can do in artificial insemination is you can up the number that are occurring in the cervix by the number of sperm that are in the uterus by passing an insemination tube through the cervix. Good artificial inseminators, very good at manipulating the cervix, passing those little plastic straws, and then injecting the sperm right into this area. That increases your probability of conception. Up in the upper right diagram, you have a picture of this. There's the ovum. There is the zona pellucida, and then this cumulus oophrus. So the whole egg mass and the ovum is moving kind of in concert. They have no way to propel themselves. What they require they require the fimbria to sweep them into the uterine tube, and then cells of the uterine tube having little smaller villi to sweep and move the egg down. So there's a lot of egg movement. The ovum may get fertilized here, it may get fertilized here, it in fact may move into the other uterine horn and get fertilized there. So it's kind of like the sperm are looking for the ovum is looking for the egg. The egg, large amounts of nutrients, particularly relative to the sperm. So if we look at a sperm, here it is, a spermatozoon. So this is an adult sperm. Remember we had the spermatogonium in the seminiferous tubules. That underwent meiosis and mitosis to form spermatids. These were kind of the immature sperm. The spermatids moved into the epididymis, and then they matured. So this is a mature sperm or spermatozoon. It is characterized by a head region, a neck region, a middle piece, and then a tail. And what you should appreciate in this drawing is, particularly when you know that the head is composed mostly of a nucleus, that there's not a lot of cytoplasm. There's mitochondria. These are located down here. Their role is to provide ATP for the tail or flagellum, because it's this motion and this movement that's going to propel this little sac of DNA throughout the uterus. The head consists not only of a nucleus but a cap, what's called the acrosome cap. The acrosome cap is a glycoprotein. It's very important in the insemination process, particularly because it contains a hyaluronidase. Recall one of the components, the sticky viscid components in the antrum of the follicle was hyaluronic acid. So here's a side view. Let's move over a little bit. There's our ovum. So that's the haploid female gamete. There's the zona pellucida. These are the cumulus oophoris cells, and this whole sticky mass is held together by hyaluronic acid. Well, if you have an enzyme, hyaluronidase, that helps degrade this, then it facilitates the movement of this sperm through this layer and towards the ovum. Okay, capacitation. There are processes that set up and make this fertilization more effective. In order for the sperm to be effective, it must undergo a process called capacitation. This is where the cap becomes loose, and there's a loss of some of the glycoprotein, making the availability of the hyaluronidase to be effective. And that's shown then in what's called the acrosome reaction. So capacitation is the preparation of the acrosome. The acrosome reaction is the release of the hyaluronidase acid. Hyaluronidase, the enzyme. Some things that affect capacitation. Well, if you're into in vitro fertilization, some of this becomes very important. Turns out that both capacitation and the acrosome reaction are facilitated by caffeine. A little shot of Starbucks in the mix. You can stimulate and increase reproductive efficiency sometimes by increasing the amount of capacitation. In a normal circumstance, you must have both you must have capacitation and the acrosome reaction to occur. All right, so what happens? You've got the hyaluronidase released. That allows the spermatid that's still moving to move through this sticky cell layer. It then penetrates the zona pellucida. And in doing so, it does at a specific protein site called ZP3. So there's a protein then that is associated with the fertilization process. The binding of sperm to ZP3 causes the Zona reaction. So now we're bringing the ovum into it. We're saying, well, the sperm has done its job. It's been effective. It's identified the ovum. It's gotten to the egg mass. It's moving effectively towards the ovum, and we're going to have fertilization. Well, one of the things you don't want is you don't want more than one sperm fertilizing that egg. That is a condition referred to as polyspermy. In polyspermy, more than one sperm penetrates the ovum. There's more than a diploid number. Those cells will never survive. Well, remember, you've got a lot of sperm competing for this. So one of the ways that you deal with this is the Zona reaction. After sperm entry and the binding with ZP3, there is an electrical and chemical change that occurs that prevents further entrance of any more sperm. So the first sperm that makes it through the zona pellucida induces this Zona reaction, and it seals the rest of the access to other sperm. We know there's an electrical signal that occurs with this. We also know that there's a chemical signal driven primarily by changes in calcium. So calcium plays an important role in this reaction. Again, in vitro fertilization, you need to know that, well, I've got to have capacitation. I've got to have this acrosome reaction. I also need to have the Zona reaction. I need to have calcium in the mix. Once that binding and fusion occurs with ZP3, then the sperm moves in and fuses with the pronuclei of the female gamete from the ovum. So you've got a male gamete, female gamete, you got the sperm, you got the ovum, they meet together, you now have the zygote. Comment about here, sperm and egg longevity and survival. I think all you need to know is that sperm have a relatively longer half-life. They may be on the order of a couple days. Eggs are very fragile. They need to be, they must be fertilized generally within 24 hours. Characteristically, what people will do is, we don't know exactly when our domestic animals are ovulating. We have an idea. We may have a relatively close idea. But we know that egg's around for a relatively short period of time, so we might, in fact, inseminate the animal a couple times. And you go, well, why is that? Well, sperm may be existing in the uterus for a prolonged period of time, but your conception rates are improved by giving a fresh new bolus. There's not a lot of energy stores in the sperm. It's mostly mitochondria, nuclear material. It relies a lot on the nutrients that come from the uterus for its support. (157). You now have a fertilized zygote. What happens? We have the ovum. It gets fertilized. We now have the zygote. This is the diploid. It undergoes a series of cell divisions. And if you take classic embryology, you will follow this development ad nauseum. I want you to know kind of some of the general ideas, the fact that we have a large number of cells that form the morula. So we go from the zygote to the morula, and then from the morula to a blastocyst. So this is kind of our progenitor. This is our developing embryo. your notes in your book, it says the young embryo is not motile and must be transported to the uterus. Well, yes and no. You're right. The young embryo is just developing cell mass, and doesn't have sperm tails, and it's not propelling itself. But there's a lot of movement of the villi in the uterus. There are uterine contractions that are occurring that move this developing embryo from one horn to the next. This may be particularly obvious if you are into ultrasound. You're ultrasounding a recent animal and you're able to detect a blastocyst or an early embryo in one horn. You come back the next day, it's no longer in that horn. Well, what's happened to it? If we've lost the embryo, and you look and you find that it's in the other horn. So there's a period of time in which this developing cell mass is moving in the uterus. It has not yet implanted, and in fact may take a week or more to implant. So there is, if we look at the cell and the development, can you take me out of the picture just for a second? Oh, here, I'll just move up. Release. Here's our cumulus oophrus. There's our nice little zona pellucida. There's our ovum. There's the sperm coming in. We have a zona reaction to prevent these sperm from entering. So now we have a fusion of the nuclei. We now have a diploid. It goes to the 2-cell. It goes to the 4-cell. And then what you see is that there is a development and a separation of this blastocyst. And there is an outer layer that is going to be called the trophoblast. Now, tropic, remember that's supportive. These cells are going to fuse with the uterus on implantation and start the supportive component of the embryo. And that is depicted here. So here's this blastocyst, and these cells are the trophoblast cells. These cells are going to fuse with the uterus, and it is this tissue that is going to become the placenta. In your notes, they talk about the chorionic villi. And then you take embryology, they'll talk about the chorion and the amnion. Well, those tissues develop from this trophoblastic layer. Trophoblast cells are important in the release of lysozymes. You've got this wonderfully nutrient surface, but it's got a thick secretory covering, mucus. You need to kind of work through that. So these trophoblast cells help in kind of drilling or obtaining access to the endometrium, obtaining access to this nutrient-rich environment. The trophoblast cells, when they fuse with the endometrium, precipitate the release of a reproductive hormone. Right? This is the formation of the early placenta or the chorion. We have identified that as human chorionic gonadotropin, HCG. Now that's a bit specious. Chorionic gonadotropins are produced in all mammals. We just have given it this tag HCG, so everyone recognizes a chorionic gonadotropin in all our mammals. Now, if this was a bovine, it would be BCG, but you'll find in all the literature they refer to HCG, human chorionic gonadotropin. A gonadotrope. Remember that a gonadotrope supports development of the gonads. Well, in this case, it helps support production of progestins. So think about it. Remember what happened failure of pregnancy, failure of implantation, progesterone levels dropped off because of failure of the corpus luteus. There was no stimulus to keep the corpus luteum functioning. Well, now this is the stimulus. It is this HCG that is going to help maintain progesterone. Without progesterones, you will lose viability of the uterus. You will lose the supportive nutrient vascular environment necessary for that embryo to grow. In fact, it's HCG that we use as an indicator of pregnancy. So I said, right, progesterone was the hormone of pregnancy. What I meant was, in order to have a viable pregnancy, you must have progesterone throughout that layer, throughout development. But you get progesterone produced every time you have a corpus luteum. So every ovarian cycle, you will have a spike of progesterone. So you're concerned. Is this animal pregnant or not pregnant? You take a blood sample, you measure it for progesterone. Progesterone is high. You go, yes, it's pregnant because that's the hormone of pregnancy. Well, all that tells you is that you have a functional corpus luteum. Now, if you come back a week later and you measure and progesterones are still high, then yeah, you probably got a pregnancy. Or maybe you just have a prolonged cycle and you need to come back in another week. Well, early pregnancy detection has focused on the fact that HCG is characteristic of a trophoblast implanting into the uterus and forming a chorion. So the presence of HCG tells you definitely you've got a pregnancy. Now, these tests are relatively inexpensive, easy to do, requires blood sample. For many of our domestic animals, the large ones, we palpate. We can feel changes in the membranes, changes in the myometrium, changes and enlargements in the uterus where this embryo is developing. And so or we can use ultrasound to visualize it. Faster and less expensive than doing a blood test. Twins, dizygotic monodies. I'm not really concerned that you focusing on formation of twins. Regulation of pregnancy and parturition. So we've got pregnancy now. We need to maintain it. How do we do that? Plate 158. Plate 158. Plate 158. Help me here. This little box is to indicate the normal ovarian cycle. And we talked about the increases in estrogens and progestins and this stimulation of LH and FSH. Well, what happens if pregnancy occurs in implantation? So here's the implantation. And what we see then is this spike of HCG. It stimulates and helps maintain the corpus luteum. Normally during this time period there would be a regression of the uterus because no progesterone. Well, now that progesterone level is being maintained high by HCG. Those progesterones help the formation of the uterus to become even more supportive, more secretory, and to develop the placenta. Now, the placental is that interface between the chorion, the chorionic villi of the trophoblast cells, and the maternal blood supply. So there's now several cell layers. And again, that species, some domestic species have lots of cell layers associated with the placenta. Primates tend to have relatively few cell layers associated with that. But this placenta then starts secreting a compound called chorionic somatotropin. So a gonadotrope supports gonads, a somatotropin supports the soma or body. We see HCG being important in the maintenance of the uterus early on. That role is taken over by, as the chorion matures into the placenta, that is transposed into another hormone called human chorionic somatotropin. Antagonizes the actions of insulin. So now think about that. It antagonizes insulin. What that means is insulin's effect are attenuated in the female. Insulin is responsible for transporting glucose from the blood into the cells. Does that make sense? Does if you want to spare the glucose for the fetus. What you're doing is making the maternal source less sensitive to insulin, meaning that glucose that she has floating around in her body are not as likely to be taken up by her cells. That makes them more available for the fetus. Consequence? Anybody see a consequence to that? A potential pathological consequence? What if the mother becomes very insulin insensitive? She becomes a diabetic. So one of the things one needs to be concerned about is pregnancy-associated diabetics. We see that in our domestic animals. We see it a lot we see it more commonly in dogs than in our food animals. Okay, so with these diabetic associated. What else might we see? Remember the sequelae to diabetes. Lots of things. Polyuria, polydipsia. Okay, those probably aren't life-threatening. But what about ketonemia or ketosis? Remember that was a sequelae or a potential outcome from diabetes? Well, you can see that in mothers as well. Big problem in sheep. Pregnancy ketosis. Lots of ketones being produced because the animals are insulin insensitive. Now, fetus still got glucose. It's not insulin sensitive, and the fetus is taking up glucose and living this great sea of plenty. Well, that works only as long as mom is okay. If she's producing lots of ketones, remember acetone, kind of nasty stuff, it'll diffuse across the placenta and kill the fetus. So this is a potentially life-threatening disease. Chorionic somatotropin stimulates development of the mammary glands. And we're going to see that interplay in a minute on another plate. We need to prepare for parturition. We need to prepare for the body for the development or the release of this fetus. Hormones that are regulating parturition. So parturition is the birth process. Now it turns out that you need to have a drop in both progesterone and estrogen at parturition. Signals for this are varied. We think in sheep and in humans, the signal for this decrease is in fact cortisol from the adrenals and the adrenals of the fetus. So when the fetus is ready and it gives a cortisol pulse, that the corpus luteum, the ovaries, stop producing the progesterone and the estrogen. And you'll see then this precipitous drop in these hormones prior to parturition. The estrogen, progesterone hormone of pregnancy, but look what's happening with estrogen later on. That it's increasing. In fact, it will increase above progesterone at the very end. Now, it's not likely driving libido. What we see this estrogen pulse here important in is sensitizing the uterus. You need to prepare the uterus for parturition. Under the influence of estrogen, receptors for oxytocin are increased by 100-fold. That means if you were to give oxytocin down here, uterus wouldn't be particularly responsive. If you give it here, much more responsive. That means the uterus is more sensitive to oxytocin. You recall the release of oxytocin by cervical stimulation. So here's the fetus striking the cervix. That sensory impulse is going back now to the hypothalamus. Hypothalamus releases oxytocin through the posterior pituitary. So FSH, LH, all that's kind of being driven by the anterior pituitary early and throughout pregnancy. Now you have the appearance of the posterior pituitary in the release of this oxytocin. Oxytocin goes down and stimulates these receptors which are under the influence of increased estrogen. Meaning, the uterus is not going to be sensitive to oxytocin unless it's had that estrogen pulse. We can also influence this, smooth muscle contraction, by the use of prostaglandins. So in some of our domestic species, we use oxytocin to help in parturition. In other, we use prostaglandins. And in your reproductive class, there will be a fair amount of detail of that. Plate 159. Regulation of mammary growth and lactation. So again, a bit of a review. We're talking about the mammary gland. Males have mammary glands. They just are not pronounced. They're also not usually under the control of the hormone estrogens and progestins. Now, it turns out that in some pathological conditions, urinary incontinence is one example. In dogs, in which we use estrogen to modify the sensitivity and responsiveness of smooth muscles of the bladder. So those dogs that are undergoing estrogen treatment to cure incontinence will, guess what? Develop more mammary tissue. So both males and females have mammary glands. It just tends in females the importance of estrogen and progesterone. In adolescence, prepubertal, as we're developing the mammary glands, there are what we call alveoli and ducts. So like in the lungs, there are these little sacs that are lined by cells. It's these cells that are responsive and responsible for the production of milk. They secrete the milk into the ducts that are then delivered through the udder or breasts into the nipples. But you need now to develop these alveoli. So what happens? Well, we talked about hormones of pregnancy, progesterone and estrogen. are critically important in the development of the mammary tissue. Particularly in the later stages, as estrogen and progesterones are increasing, mammary glands develop massively right prior to parturition. So you now have this structural framework. What you need is you need to have prolactin brought into it. Prolactin is under the control of prolactin inhibiting hormone and prolactin releasing hormone. You recall those are, prolactin is released from the anterior pituitary. The inhibiting and releasing hormones are from the hypothalamus. So again, we come back to the hypothalamus, anterior pituitary, preparing this tissue for milk production. Human chorionic somatotropin, development of breasts, are also under the influence of HCS. These cells are going to be milk-producing cells. They're going to be using a lot of glucose. So, in fact, we find that insulin plays a role, as does glucocorticoids and growth hormones. Glucocorticoids, growth hormones, important in growing the tissue, preparing the tissue for glucose metabolism, and you're kind of like gearing this whole system up for the delivery of milk, all occurring in the later stages. So when we look at our domestic animals, they'll start bagging up. The udder, mammary glands are getting larger. You may see some milk production, a little dripping occurring right prior. In fact, that's one of your signals. This animal is about ready to deliver. But remember that milk ejection, or what we call milk letdown, requires oxytocin. You don't get oxytocin until delivery, and stimulation of the cervix, and remember also stimulation of the udder itself. And you see that down in this graph down here to remind us. Here's the hypothalamus, anterior pituitary, posterior pituitary, prolactin being produced, but it's this sensory stimulus that comes back, feeds back onto the hypothalamus to cause the release of oxytocin. Prolactin milk formation, oxytocin milk ejection. Just a couple comments about composition. Again, for this class, you don't have to know different compositions. This is strictly comparative. But some of the elements that we measure and are concerned about when we're delivering milk is the amount of carbohydrate. The primary sugar of milk, you should know, is lactose. Sugar. Of course, calcium. Got to have calcium, right, for everything else. Protein and lipid. And from species to species, these numbers may vary incredibly. You, who might be involved in animal rescue, have to know about these differences. Feeding an animal too much lactose, if they don't have adequate amounts of lactase, can lead to diarrhea. Similarly, if you're trying to raise sea mammals on 3.8 and 3.7% fat, they're not going to get enough nutrients. Initial milk. So I need to make a comment about the initial formation of milk that distinguishes it from like 3 or 4 days out. The initial milk that's produced has huge concentrations of antibodies, at least in some of our species. So dogs, cats, humans, a lot of antibodies are transmitted through the placenta prior to birth. So we are born with relatively large concentrations of immunoglobulins, immune proteins circulating in our body. That's not true with many of the food animals. Food animals have more cell layers than their placenta. It makes transport of proteins from the maternal source to the placental source more challenging, and as a consequence, these animals may be born with zero or no immunoglobulins. Well, where do they get that? They get it in the first milk, and the first milk, colostrum, is antibody rich. It is critically important that these animals get those antibodies. Failure to get the first milk, failure to get those antibodies is referred to as failure of passive transport of immunoglobulins. You're not getting them. You're not getting them from the milk. That makes that neonate much more susceptible to disease. So even in dairies, right, you've got to have a parturition in order to get milk production going. You want to make sure that those young calves that come off their mothers get adequate amounts of colostrum. You don't want to have to fight a disease process them with the youngsters because they're no longer around mom. Mom should give her first colostrum to the calves, and then she needs to produce milk to make money. And I think we should always stop talking about money, but let's stop there if we could.